Good morning, everyone. Today, I'll be talking about some aspects of frugivory and seed dispersal by an important group of birds, the hornbills. And I am Aparajita Dutta. I'm a scientist at the Nature Conservation Foundation. I've been working in Northeast India for about almost 18 years now, and mainly in Arunachal Pradesh. So today, I'll be talking about a work that has spanned about 15 to 18 years in trying to understand frugivory seed dispersal and secondary seed dispersal and seed predation in the tropical forests of the Eastern Himalaya. As all of you know, plant-animal interactions, there are a number of different kinds of plant-animal interactions, some of which are antagonistic, between the players, and there are consumer and resource interactions like predator-prey interactions, as well as competition. Some of these are antagonistic, but there are also mutualistic interactions like pollination and seed dispersal. So my talk will focus on seed dispersal. Seed dispersal is what is called a dispersive mutualism, because it involves the dispersal of seeds in return for the nutritional value of fruits or other structures that's associated with the seeds. So the plants benefit by the seeds being carried away from them, and they produce these fleshy rewards for a variety of dispersers that carry these seeds away. And so the dispersers or the consumers are also getting nutritional benefits. So why is seed dispersal beneficial? In the sense that why can't seeds just germinate below the parent plants? So there are several hypotheses related to why departure from the parent plant is useful and beneficial for ultimate recruitment and seed germination and ultimate recruitment. And this is because of certain things called distance and density dependent effects around the parent tree. So because seeds are in high densities around parent trees where the maximum fruit fall happens, there is a high degree of different players that affect the seed survival. So there will be pathogens, there will be seed predators. At a later stage, out of the seeds that escape mortality, there will be seedlings that are attacked by herbivores. So there are a number of different factors that affect germination and recruitment under parent trees, which results in, so it's the escape hypothesis, basically that seeds benefit from being carried away from the parent trees. Seed dispersal also has what are called these arrival related benefits, because seeds are carried to, and they're non-exclusive, some of these hypotheses. So they get carried to favorable sites and microsites for germination and recruitment, where dispersers are carrying the seeds away to particularly favorable sites, for germination. And this has been demonstrated in some studies, in South America, with a particular species that takes the seeds to these open perches in canopy gaps, which are beneficial for the survival and the germination of seedlings. There's also the colonization hypothesis. So seed dispersal is also beneficial because there are sometimes ephemeral, unpredictable new habitats created, vacant habitats, which the arrival of seeds there allows plants to regenerate and expand their area of occupation. Also in new volcanic islands which have been destroyed, there is a famous example in Indonesia, in Krakatoa, of the recolonization by plants after it was, after a volcanic eruption. There's also latitudinal migration in plants in response to climate change. So the main, one of the main hypotheses that drives a lot of the seed dispersal research has been this hypothesis, which is now known as the Jansen-Connell hypothesis or model, and it was proposed almost simultaneously by Dan Jansen and Joseph Connell. And basically it posits that as you move away from the parent tree, while the densities of seeds are high near the parent tree, as you move away from the parent tree, the probability of seed dispersal, the probability of seedling survival increases. So, so there's lots and lots of seed and seedling mortality near the parent tree, but as you move away, there's less seed and seedling mortality. So, this hypothesis has been the basis for a lot of the research. So, seed dispersal, as I've said it already, is that movement of seeds from parent plants. But seed dispersal can also be mechanical, in which case it is not a plant-animal interaction. So, a large number of species are also mechanically dispersed by various agents like wind, water, gravity, and through explosive mechanisms. There is also animal dispersal, obviously, which is known as zoochorus. And so the way the dispersal happens can be externally on the body fur of many animals. Like in this picture, it shows a goral, carrying seeds of a particular plant on its back inadvertently. And also the most important is the endozoochorus, or the internal seed dispersal. That is basically when frugivores ingest the fruits and they either regurgitate or defecate the seeds out in different sites where they germinate. So, especially in tropical forests, 90% of tropical woody species produce fleshy fruits, and vertebrates are among the most important seed dispersers, and almost 85% of woody species are dispersed by birds and mammals mainly. Fishes and even reptiles have been the focus of an increasing number of studies, reptiles such as turtles and lizards, and even fishes, especially in the Amazon, have been found to be major dispersers in some of these river kind of ecosystems. There's also certain invertebrate groups like ants and beetles, which have been studied extensively and shown to be important seed dispersers. Secondary seed dispersal is another step in the process, by which what we mean by secondary seed dispersal is that after the primary dispersal by the main frugivore or disperser, sometimes seeds get taken away by rodents to other places where they are cached. So that is known as secondary seed dispersal, because it's another, it's moved away from the original site of dispersal. And usually in the case of rodents, not usually, always in the case of rodents are seed predators. So they will, their main aim is to eat the seed. But because they sometimes cache it in a place and forget about retrieving it, they are doing secondary seed dispersal by removing the seed and leaving it somewhere else. So the seed dispersal cycle, this is the whole idea of the entire process of seed dispersal involves a lot of things. First, there are multiple aspects to it, and it starts with fruit production, and then fruit availability, and then there is fruit removal by frugivores, and then there is seed uptake by frugivores, and after that, so fruit removal is affected by a lot of factors, which includes diet choice of frugivores, nutrition, visitation rate, fruit removal rate, and all that. And then the seed is taken in, the seed is either regurgitated or defecated out depending on the frugivore, and then seed dispersal happens. Seed dispersal is also then dependent on multiple things like gut passage time, the movement of a disperser, and also if it is being secondarily dispersed, whether it's scatter-hoarded and whether it's cached by various secondary seed dispersers. The seed dispersal creates a kind of template on which seeds are arriving, and this is like seed rain or deposition of seeds. After this is the germination stage, And here there are a lot of different factors that affect the mortality or the survival of these seedlings. That then determines the seedling distribution, and then there is a recruitment phase, and much research has shown that between the seed to the seedling and the seedling to the sapling stage is where the maximum mortality occurs. And so the sapling distribution is also determined by some of these density dependent mortality factors, and ultimately the saplings come to the pole stage or the adults. Now the problem, as you can see, is that for a single study to be able to follow this full cycle is impossible, especially in the case of trees, which have very long life cycles. So much research in Frugivory seed dispersal has focused on different aspects of the cycle, many studies, and in more recent times there have been long-term studies that have tried to incorporate all aspects, or at least some of the aspects. There's also been a divide in which a lot of people who focused on the frugivory or the animal part is different from the people who have focused on the seedling and sapling recruitment and the plant part. So establishing the loop, closing the loop is very, very difficult to do and challenging, because to demonstrate the effectiveness of a seed dispersal, of a seed disperser, you would ideally have to go through most of these stages, to be able to show how effective a disperser is. And most people just end up showing part of it. So coming to our research in the Eastern Himalaya, our main study sites have been in the Pake Tiger Reserve, which is in western Arunachal Pradesh, and also in Namdafa National Park or Tiger Reserve, which is in eastern Arunachal. And we first began work in Pake in '95, and we began some of our work on hornbill seed dispersal from '97 onwards, and we've been continuing to sort of try to put together a full picture of the role of hornbills. So in the initial phase, when I started my PhD research there, one of the things that I documented as part of my work was that out of around 165 tree species that we documented in our vegetation plots, 78% of the tree species in Pake Tiger Reserve were dispersed by animals, and bird dispersal was even more prominent than mammal dispersal. So fruits were adapted mainly for animal dispersal. And there are a number of different frugivore groups that are found in the Eastern Himalaya, which includes many species of civets and the binturong, the yellow-throated marten. It also includes deer. There are several species of deer like the barking deer and the sambar, which are also important frugivores at times, and they disperse seeds. Of course, there are the hornbills, and there are a number of other frugivorous bird groups such as bulbuls and barbets and imperial pigeons and mynas. There's also bats, which are really poorly studied in India, at least especially in the northeast. Then, and there is also this phenomenon of secondary seed dispersal by rodents. So when I began my work, we didn't know all aspects of who are the dispersers. Our main study question was to look at how effective are hornbills as seed dispersers. And this picture, by the way, is not of an Indian species. It is a picture of critically endangered rufous-headed hornbill, or the Walden's hornbill, found in the Philippines. And so my initial research on the diet of hornbills showed that fruits comprised 90 to 95% in the diet of hornbills, and they were dominated by two types of fruits mainly, figs and non-figs. And ficus is very important for hornbills. And among the non-figs, you had these fruit types called drupes, which are these fleshy single-seeded fruits, which are usually black in color, and the arillate capsular fruits, which belong to certain families of the Meliaceae, the Myristicaceae, and the drupes also belong to the Anonaceae and the Lauraceae, Myrtaceae. So, during my research, in the 4 years of my PhD research and subsequent years, we have overall documented about 100 fruit species in the diet of hornbills. And as you can see from the images, hornbills seem to prefer fruits that are black in color when ripe, or they prefer fruits that are a combination of black and red, or orange, those kind of colors. And the hornbills also do not, at least especially the larger hornbills, do not eat unripe fruits at all. So their aim is to eat the soft and fleshy part of the fruit. And some of these fruits, like the Horsfieldia kingi and the Nema angustifolia and Aglaya from the Myristicaceae and the Meliaceae families, are these arillate capsular fruits. So they dehisce on ripening, and the hornbills eat the aril, the fleshy aril. The other fruits like the Polyalthea and the Livistona are these drupe-like fruits, and of course there are figs. So one of the things that we showed is that hornbills are really large. As most of you might know, hornbills, the weight of these larger hornbills is 3 to 4 kilos, and they have a very large gape size. And gape size determines the ability of birds to the extent to which they can swallow the size of the fruits that they can swallow. And given that hornbills have very large gape widths, our work has shown that the 3 species of hornbills in this graph, you can see that they stand out as the main, as the 3 frugivores that swallow the largest diameter of fruits, compared to some of the other frugivores which only are able to consume fruits which are below 2 millimeters. The other, 20 millimeters, 2 centimeters. The other 2 species, like imperial pigeons, also feed on fruits which are, they tend to feed on fruits which are larger than expected based on their gape width, and so is the great barbet. So one thing you have to remember is that hornbills do not, while they can eat the smallest fruits also, they can also, they are the only ones who can swallow whole the largest sized fruits. So seed disperser effectiveness, how is it measured? There are 2 different aspects to it. One is the quality part, and the other is the quantity part. How many visits are made, how much fruit is eaten, incorporates, how many seeds are dispersed, is the quantity part. Qualitative part includes a number of things like fruit handling behavior, then the visitation length of the bird at fruiting trees, and I will explain why that is important later, gut treatment of seeds, the viability of seeds and whether they germinate well after going through the gut of the animal, then the time taken to move through the gut, and where are the seeds deposited, the site of deposition, whether the site is suitable for establishment, and also the distance from the parent tree, as I had pointed out earlier. So there are a number of factors, and this was first proposed by Eugene Schupp in 1993, and he further expanded it and improved the idea of this in a later paper. In the I talked about the quantitative part before, but in the quantitative part you have frequency of visitation and the number of fruits removed per visit. So in the first part of this presentation, I will talk about the first 3 aspects of quality by hornbills. So one of the things we looked at is what proportions of fruits handled by hornbills were swallowed, And what is the visitation length on fruiting trees? So if visitation length is greater than seed retention time, that is not good, because the bird will spend a long time sitting on the parent fruiting tree and likely will not be taking the seeds away from the parent tree, because the seeds will be deposited under the parent tree because they are sitting longer than the gut retention time. If the visitation length is shorter than the seed retention time, then they are good dispersals, one aspect of the quality. So what we found is that hornbills mainly tend to swallow fruits. Compared to the other two frugivorous groups, avian frugivores and imperial pigeon, hornbills swallowed fruits much more than they either dropped or pecked, whereas the other frugivores tended to drop them while foraging or pecked at them. As you can see, hornbills are very picky eaters, so they will, when these fruits dehisce in these arillate capsular species like this Disoxylum, the hornbill will, test the fruit's quality and softness and then swallow it, and it gets processed in the proximal gut and then it's regurgitated. Most large seeds eaten by hornbills are regurgitated and not defecated. Only ficus, which has minute seeds, are defecated. The other aspect of quality is how long you spend at the fruiting tree. So if you spend too long at the fruiting tree, that's an indication that you may be ending up dispersing the seeds under the parent tree. So in the case of hornbills, as you can see, the visitation length is around less than 10 minutes. Whereas, you can see that the imperial pigeon, which might be a good disperser on some counts, but in terms of the length of time it spends at a fruiting tree, it's pretty long, and it can potentially end up dropping the seeds below the parent tree. One of the other things that I did during that time of my research was look at whether germination of seeds regurgitated by hornbills was better and more than germination of fallen seeds which are collected from under the parent fruiting trees. So I did this for 37 food species. I also first tested for seed viability, and I found that regurgitated seeds are viable and germinated in 18 of the species, and germination success of regurgitated seeds varied from 13 to 90%, and for 5 species there was higher germination success when they were regurgitated by hornbills. For 7 species there were no difference. These were with chi-square tests, and for 7 species they failed to germinate, but that is probably because of very small sample sizes. The other thing that I did in that time was there were 2 captive hornbills in my study site, so one great hornbill and one wreathed hornbill. So I fed them fruits of ficus as well as 13 different non-fig food species, and overall I found that the gut passage time for, I'm not presenting all the different gut passage times for all these different species, but basically it was around 75 minutes or more than 1 hour, and the defecation time for figs was around an hour. So in a subsequent study done by some of my researchers in our team with about 6 captive hornbills, we looked at the regurgitation times for 5 large seeded and medium sized tree species, and we found again that the range of gut passage time is quite, although the range was very high in this case, in this study, but the median gut passage times were pretty high, more than an hour, in fact, even 2 hours. So, so as you can see that I have talked about these aspects to show that hornbills are handling fruits well. They have short visitation lengths. Their gut treatment makes seeds, leaves seeds viable, as well as in some cases helps germination. So now I will focus on the next aspect of quality, which is where is the site of deposition, site suitability for seed establishment. Now remember that when people did research on Frugivory seed dispersal, people in earlier studies often stopped at the first 3 aspects when they were looking at the animal side of things. people generally did not look at the deposition sites, and the suitability. So hornbills disperse seeds in 3 different ways, or 3 different sites. One is when they are foraging in the daytime, they just travel and move from from the fruiting trees to perch trees, and they will disperse seeds at perch trees, where they spend maybe a shorter amount of time, and where it might be single birds or pairs or a fewer number of individuals. The other place where hornbills disperse seeds is at nest trees. Now, hornbills have a unique breeding biology, which many of you might be aware of. They seal their cavities. The female seals themselves inside and is fed by the male. The female and the growing chick is fed by the male throughout the long breeding season, which is almost 4 months for the larger hornbill species. So during that time, when the female and chick is inside the nest, the male is feeding the female and chick, and all the seeds that the female and the chick are eating, the fruits that the female and chick are consuming, the seeds are regurgitated out of the nest cavity, and they fall within a small area right below the nest tree. So in that instance, hornbills are doing clumped dispersal. Similarly, when hornbills have this behavior of roosting in large flocks at the end of the day, and they would gather, especially the wreathed hornbill, some of the species roost in very large flocks of up to 100 birds or more in the areas that we work in, and this can range from 5 to 10 birds to 100 birds at different times of the year. But when they're roosting, they use a site continuously over a period maybe up to 2 months, and a few trees in that site. So a huge number of seeds get deposited under these roost trees. So there are 3 types of deposition sites. So at nest trees, hornbills are doing clump dispersal. At roost trees, they're also doing clump dispersal. But when they are foraging in the daytime and moving around on perch trees and fruiting trees, they are scatter dispersing. So one of the one of the plots we did, some of the plots around trees we looked at the seed densities to show you that the densities of seeds are way higher under roost and nest trees compared to perch trees, where hornbills spend shorter periods of time and which are more unpredictable in space and time, and which are, they are moving around. So as you can see that hornbills in the roost sites, seed densities are up to 44 seeds per square meter. At nest sites, it can be 100 seeds per square meter, whereas at perch sites, it will be less than 1. So there's concentrated seed rain, and during the 4 years that I studied the breeding biology in the early 90s, I found that up to 27 species make up the seed rain, which is massive, at 27 nests, and there were and ultimately you see that there's a variability in the, even though 27 species were in the nests under the nest trees, the seed rain, the seedlings, ultimately the seedlings that came up belong to a much smaller set of species, and this is because of differential survival of these seedlings, which are due to a number of factors, including seed removal and seed predation, which I will not speak about now. So one of the things that we did is look at, okay, there's a lot of seeds being deposited under nest trees. Is regeneration happening better, where the hornbills are, So we checked the regeneration under nest trees front and back of cavity. So back is like a control, no? So the front of the cavity is where the seed drain is happening, and the back. So you find that, yes, there was a high seedling density of hornbill food plant species in the front of the cavity, and very low at the back. And conversely, like when you have non-food species, there was no difference, which is what we expected. So yes, there is regeneration. But if you look at the sapling stage, you find that these differences disappear because there is further mortality. Okay, yeah. So at roost trees, there is also a huge amount of seed rain. 115 seeds per day is my estimate, and like I already said, high seed densities. But when you look at the regeneration, you see that even though seedlings come up initially and they are high seedling densities, the sapling density is drastically lower and tree density is almost nil. And this is because hornbills roost in open sites away from the main forest patches where they forage along the river. They roost along the river on these tall trees of bombax and albizia, where these are kind of semi-grassland kind of areas where there's inundation, there is other disturbances that ultimately does not result in recruitment anyway. Under perch trees, I'd already indicated that the number of seeds deposited are much less because these are more unpredictable in space and time. They're visited for shorter periods. So so that was it. And then we just showed that why is it bad, this kind of clump dispersal. So we compared the seed predation rates at parent trees versus perch trees and found that perch trees had much lower rates of seed predation by rodents. And the rodent seed predation is very obviously detected by these marks, tooth marks. We also looked at seedling survival, which is the next stage, which very few studies look at. And we tagged the seedlings under parent trees. We tagged a subset of cohort of seedlings under nest trees and in both years, in 98 and 99, and in under roost trees. And we found that seedling survival is, very, very poor, even at nest and roost trees, and it's as low as under parent trees. Okay, so but a subsequent study by so we had not in this study we had not looked at the what happens when hornbills scatter disperse seeds in the daytime during foraging. What is the kind of deposition rates and all that? And one of my PhD students, who is now my colleague, Rohit Nanewadikar, he studied the scatter dispersal of by hornbills in the Namdapha Tiger Reserve, and he found that hornbills disperse a huge number of seeds per day per square kilometer. I will not present the results of that work here. So as you've seen, I have talked about the various aspects of the quality. One of the remaining aspects is distance from the parent tree, and of course the quantitative part. So distance of dispersed seed from parent tree is the most difficult thing to assess, in tropical forests and especially for large frugivores like hornbills. And how you do it is by incorporating the regurgitation times of seeds which you have taken from the gut passage experiments, and by understanding fine scale movement. So that you can do only if you can collar and tag, if you can catch the hornbills and tag them and track their movement. So we did that. And we put these, GPS tags on these hornbills. I won't go into the methods right now, but basically what we found is that this map shows the movement of a great hornbill, just depicting that the hornbills know no boundaries. They are moving from the tiger reserve to the reserve forest and into Assam. This area is at the border of Assam and Arunachal Pradesh. And as you can see, hornbills are dispersing seeds also through these movements into areas which are more degraded. The wreathed hornbill moved even more than the great hornbill. And this table basically shows we captured 6 great hornbills and we estimated their home ranges. And the great hornbill home ranges in the breeding season was very, very small, mostly around 1 square kilometer, 1 to 2 square kilometers. And the great hornbill home range during the non-breeding season from 2 birds is between around 60 square kilometers, whereas the wreathed hornbill home range, even in the breeding season, where you'd expect them to move less, was 54 square kilometers. Unfortunately, we could not tag wreathed hornbills for the non-breeding season, and wreathed hornbills are supposed to have much more wide-ranging movements. So we got the mean daily distance moved and all those, and which indicates that hornbills move quite a bit, especially the wreathed hornbill. Okay. Now, how do you get from all this movement data and the gut passage time to the understanding the distance of dispersed seeds? So we knew the GPT of those seeds that we had passed through the hornbill's gut in those experiments, I think around 305 seeds. So and we also had from our data on the movement, we used some random start location based on the foraging distribution of foraging observations. And by using the start point, and every seed was assigned, like the gut passage of every seed was assigned to the random starting point and the end. So the displacement gives us the amount that the hornbill could have moved, the probability of the seed being dispersed to certain distances. So this is the way you integrate GPT and movement data, so to determine the final location. So quickly, I just want to show that the great hornbill breeding, during the breeding season, the great hornbills median distance of seed dispersal was 294 meters, and the maximum distance was 2.5 kilometers, which is quite high, I mean, based on other known seed dispersal distances of other frugivores. For the wreathed hornbill, which is in the gray bars, there was a bimodal kind of seed dispersal distribution, where there was a peak at around 500 to 1 kilometer, and there was later another smaller peak at between 4 to 4.5 kilometers. The median distance was 1.3 kilometers, and the maximal distance seeds could be dispersed was 10.8 kilometers. Similarly, for the great hornbill in the non-breeding season, the median distance was pretty similar to the median distance in the breeding season, but in the non-breeding season, the maximum distance went up to 12.8 kilometers. So, as you can see, the wreathed hornbill, tail of the distribution is very, very long, and which means there's a lot of, there's some seeds that really go very far, up to 14 kilometers. So I have established the quality aspects. Now the quantity aspects. So one of our other students, he did a study looking at the important hornbill food plant species and observed 8 of those fruiting trees for many hours, and he quantified how many birds came, how many fruits were swallowed. And what he found is that among all the visitors or frugivores, there was hornbills, there were small avian frugivores, there was imperial pigeons, squirrels, and primates. But hornbills stand out on the top right hand of the graph, where you can see that not only was the visitation rate on the x-axis high, but so was fruit removal rate, which was not the case for the other groups. The same thing is shown in this graph, which is basically the visitation rate on the left hand side, and shows that hornbills have although the hornbills have lower visitation rates than imperial pigeons, the number of fruits removed per visit adequately compensates for that. It's more than double or triple the number of fruits removed by imperial pigeons. So I have established that a number of factors, make hornbills good dispersers, but that given the types of sites at which they deposit when they're doing clump dispersal at nest trees and roost trees, there's a lot of mortality and high levels of seed predation and seedling mortality, which sort of mimics what happens at parent trees. But hornbills are indeed farmers of the forest, because they scatter disperse seeds, to a large extent when they're foraging during the daytime. They move large distances, and the distances of seeds moved by them, by both the larger species, is quite high. And so therefore, this study has basically looked at all aspects of the whole gamut of establishing the effectiveness to understand the role of hornbills as seed dispersers. So moving on from hornbills, after working for so many years on hornbills, in 2011 or 12, we also wanted to look at the role of rodents, in this system. Because as I indicated in parts of my talk very casually, was that there was seed removal or predation at nest trees, or so for me that was a question that remained in my mind. Are they mostly removing it, or are they predating it on site, which means those are gone. But if they are removing it and caching it or taking it away, there is a chance that rodents are sort of changing the template of seed dispersal created by hornbills by thinning out the seed densities at nest trees and taking them away. About secondary seed dispersal has been studied very well in the neotropics. It's beginning, it has, there are a number of studies from even China, but in the tropical forests, in the paleotropical forests, the role of rodents in secondary seed dispersal is very, not so well known. And there are a number of terrestrial rodents in Pake Tiger Reserve, and basically in the Eastern Himalaya. There are a large number of species, and many of which have not even been properly taxonomically identified properly. But so we wanted to know whether there is some kind of caching happening in this system also. And so we set up a study using camera traps to monitor the fate of these seeds by tagging seeds. And some of the main rodents in our study system are the Himalayan crestless porcupine, the hoary-bellied squirrel, which is an arboreal diurnal squirrel, but I will I'll show you that we found that it was also coming to seed plots. And there's some murid rodents like Niviventa species and ratters, and there are some other ones which we haven't fully identified properly, as well as the brush-tailed porcupine. So the brush-tailed porcupine and the Himalayan crestless porcupine are history seed rodents, whereas these are the murid rodents. And we chose 10 tree species, and these are the seeds of those tree species: Terpinia pomifera, Taloma hodgsoni, Actinodaphne obovata, all these, some Lauraceae, some of these Anonaceae, and Canarium, and Prunus. And basically they differed in, by the way, this is not scaled to actual seed sizes, but and so these range from very small seeded species to large seeded species. So what we found, we tagged about 10,777 seeds, and we found that rates of seed removal was much higher than direct on-site predation. We could not trace the fate of many of the tagged seeds, so the data was limited to around 4,000 seeds ultimately, I think. But even, so there we found that 44% of the seeds were removed, and they were not, on-site seed predation. And our camera traps showed that while the two species of murid rodents removed and cached seeds, they took the seeds away from the seed plot, while I didn't tell you, but we tagged seeds with these nylon kind of threads, which are up to 40 meters in length, and you can find them. So in the habitat. This is published in a paper, so you can look it up. But anyway, the two species of porcupines, on the other hand, only preyed on the seeds. They just ate up the seeds on site where they found it. And the most novel finding that we made during this study was that, which has not been documented before, was that a species of cricket, was caching the seeds of 3 species underground. So we noticed that, some of our seeds were disappearing, but we were finding these tags above ground, and if you dug deep, and we also found these dead crickets underground. And our field staff also had observations of seeing these crickets in their agricultural fields, removing material and putting it underground. So we didn't know really what was happening. We could not get the cricket on a camera trap, but we could see the live crickets, as well as we saw these dead crickets underground with the seeds. And they were also germinating underground, but that's comes later. So the rodents and cricket made 309 caches, and over 40% of the seeds were recached from the primary cache locations. So studies in the neotropics have shown that rodents not only take it at one time, they do multiple caching. Like they will move the same seed multiple times over, and ultimately the dispersal distance of the seed from the original site might be quite high, So, and we also found that about 12% germinated in the primary caches. So this graph just shows you the visitation rates of porcupines and murid rodents on our camera traps on 7 of those tree species. And as you can see that murid rodents visited almost every species, and porcupines were much less. Seed handling by rodents. So this shows porcupines handled a few of the seeds of Chrysocheton and Prunus selanica, which they predated completely, while murid rodents removed or cached seeds of the other species as well as Prunus. If you look at the fates of seeds in the caches, you find that the tree cricket removed 57% of the seeds, and in that there was these different fates. For murid rodents also there was removal, and the hoary-bellied squirrel was also removing seeds. So these are the different cache owners, and that's the cricket which we found. So we looked at 489 cache seeds. So most caches were single seeded, and this is what is called scatter hoarding. It's not like in the in the temperate forests you have pine forests where woodpeckers or some of these squirrels do larder hoarding, right? Whereas here it's like scatter hoarding, which is more common in these tropical forests. And the seeds were moved maximum distance of up to 19 meters, I think, in the case of the squirrel. And if you can see that murid rodents, the distance of a cache seed is around 5 meters median distance, a little less than that. But the problem was that our method, we could, in some cases we were able to trace the seed fate after the second level of caching, but beyond that we only got third level of caching in a few cases. So we do not know the end distance for many of the seeds, but we do know the primary level of caching. So but the cricket did not move it very far, but it was burying them underground. So this is the strata of the kind of strata in which caches were found, and the seed fate based on those different strata. So there were underground caches which included seeds cached by the cricket. As you can see, they were like in the underground, and there were like many of them were intact and germinating also. Whereas in the case of leaf litter, which was most of the seeds which were in the leaf litter, they were cached by rodents and hoary-bellied squirrel. There were some caches in the tree hollows and other kinds of substrates, mainly by murid rodents, where a lot of them ended up getting eaten ultimately, So in the leaf litter, a lot of them got further removed. So the role of rodents in secondary dispersal, what we found in this system is that murid and hystricid rodents play different roles. Porcupines are only seed predators, while murid rodents are removing or caching them. And one of the new findings is that there was dispersal and caching and burial of seeds by a true cricket species. And based on this study, we can say that removal and caching and recaching was common in this system for most of the tree species by the murid rodents. Most of the tree species, we of course studied only 10 tree species, but all these 10 tree species were being hoarded and cached by murid rodents to some extent. So to conclude, I'd like to say that in the Northeast, a lot of studies, there have been other studies which have looked at other aspects of seed dispersal and the effects of logging and hunting on seed dispersal and plant communities. And but I think a lot of new groups and a lot of research questions need to be answered about the role of bats, the role of primates, the role of some of the civets in seed dispersal. It would be wonderful if there was more research on this fascinating subject in the Northeast. Thank you.